Now we will understand another schematic representation. This is very important compared to the three schematic representation. That is action of a specific restriction enzyme. I have mentioned it. EcoR1. Got it? So this 1 is nothing but Roman number 1. That is nothing but the order of extraction of a specific type of restriction enzyme from the E. coli bacteria. The order. What is the order? In this case, this is the first restriction enzyme which is extracted from the E. coli bacteria. Got it? The order is number 1. If I write EcoR2, that means the order is second. 3, 4, 5, now the number goes like that. Got it? Okay. Very important. No confusion, nothing else. Please understand carefully. Okay. Listen to me carefully. Now, what I have to do is I have to draw the vector DNA here. So, vector DNA. The diagram now differs, but the procedure is same. It's a basic. Okay. So, I have to go with the pink color itself. I am going to draw the vector DNA, but in this case, for our understanding, I am going to draw the linear vector DNA. Okay, normally they are circular, but for our understanding, we will go like this. Okay, so here what I have to write is I am going to explain about the action of this particular EcoR1 restriction enzyme. So their recognition site or restriction site is GAATT, that is nitrogenous bases, 6 nitrogenous bases. Okay. And it is a palindrome also, so we just write C T T A A G. Done. This is actually the sequence which is recognized by this particular enzyme. So there are so many different types of restriction enzymes. Their recognition or recognition site or restriction site also varies. Got it? So for, let us say, HIND number 2, recognition site is entirely different. It will not work on this particular recognition site. So only EcoR1 will find or identify this particular palindromic sequence and cut. Okay, here I have to use a blue color to draw the DNA, which is foreign DNA containing gene of interest. Okay, we just write foreign DNA. We just write this. And the sequence, because I'm going to treat this particular DNAs, 2 DNAs with the same type of restriction enzyme. That has to be the procedure. I should not change the restriction enzymes according to my wish. Okay. So I have to write GAATTC. In this case, CTTAAG, like this. Same sequence. So the EcoR1 will identify this sequence on this sequence present on 2 different types of DNAs. That's it. Okay. So what's going to happen now? The action. Now I have to treat this particular DNA with EcoR1 type of restriction enzyme. Their point where exactly they cleave the DNA. So I have to use white color so that it will be easy for you to understand. Okay. Exactly here. So on one strand they are going to cut the DNA between G and A. Got it? And the other strand they are going to cut the DNA between G and A. But exactly opposite. See, G and A. In this case, G and A. Got it? Like this. Not here. So one strand G and A, the other strand G and A. Here also same. Again, the cleavage point is G and A for EcoR1. Again here the same. That's it. Got it? Once I treat with the same restriction enzyme, I'm going to get a DNA which looks like this. Okay, we just draw the DNA like this. This piece is now being removed. Got it? So I have only G on particular strand. Okay, so this piece is removed because of the action of this particular enzyme. And here on the other side, other strand, I have to retain C, T, T, A, but this G is removed. Got it? Why? Because of the action of this particular enzyme. So this is the thing. Hope you understood it. So to understand it, so we just put a box. That means this is gone, removed. Okay. In this case, again it's very easy now. This piece is now removed. Got it? Removed. So what I have to do is now want to draw the diagram which containing, white color, T, in this case. Here I have only G. This portion is now removed. These sequences are removed by the same enzyme. Okay, because their action is same. Oh, blue color, sorry. Okay, fine. Easy, right? Easy to understand. Done? Okay, now see here. Some sequences are missing here on one strand, and here only one sequence is missing, that is one nitrogen base is missing on the other strand. Applicable to say this particular foreign DNA also same. Okay, but can you join them? Yes, right? We can easily join them. See, missing portion, it is here, and whatever missing here also present here. So it will be easy for us to join them. That is called as ligation. So we just write step number 1, step number 2. So here actually we have to ligate them, that is join them with the help of an enzyme called as ligase enzyme. Ligase enzyme, you know about it? Okay. And this type of sticky end, see, it is unequal. One strand is short, other strand is longer. In this case it is longer and this is shorter. So it is unequal. Two unequal strands are now being produced because of the action of the particular enzyme EcoR1. Okay, got it? So this is actually called as sticky ends. So please understand it, sticky ends. Many students get confused with the sticky ends and blunt ends. Both are entirely different. What is the meaning of sticky ends? See, for the sticky ends, no need of extra procedure. Very simple, just add this enzyme ligase between the two type of enzyme, vector DNA, DNAs, vector DNA as well as this particular foreign DNA. What they do is they just join them. Let me just show that here. Okay, so we just write G here, C, T, T, A, and I'll just bring this G because of the action of the ligase. They have been joined now, and here I have write GAATTC. Done. Okay, I have to use blue color for the foreign DNA sequence. See, this is now RDA. Done. I have joined the two different types of DNA with the help of the ligase. So I have got, because of the action of this particular specific enzyme, I have got sticky ends. Okay, sticky ends are easy to join, no extra procedure is required, and I will have to write one more word here. What is the other name of sticky ends? It is also called as cohesive ends. Got it? Sticky ends can also be called as cohesive ends. Cohesive ends is very important actually. It is not called as blunt end. Note this point. We will go with one more, like I'll explain the blunt end. So this is enough. You get this question. Explain the action of restriction enzyme EcoR1. Very important. Even the same question was asked actually earlier in the board examination. You need to draw this diagram. Okay, with bit of explanation. In the schematic representation itself. No need of extra information. Okay, only simple, easy. Okay, we just write we talk about the blunt end. For that I have to take one more example, that is small 1. A type of restriction enzyme. Okay, so this small 1 actually recognizes a palindromic sequence. So we just write the vector DNA, which reads like this: triple C, triple G. That is the palindromic sequence which is recognized by the small 1. And here the opposite, triple G, triple C. Okay, and the opposite side I am using the blue color for vector. This one, what you call the foreign DNA. Again I write triple C, triple G, and in this case I write triple G, triple C. Okay, so we continue further like this. This is foreign DNA and this is the vector DNA. Easy, right? Now the small 1 action is entirely different compared to the EcoR1. Instead of cutting here, they cut exactly here. So this particular exactly equal. Exactly means in this particular strand they cut between C and G, on the other strand, the opposite strand also between G and C. So this particular portion is now being removed. Okay, on the foreign DNA, their action is also same. So they are going to remove triple C and triple G. Okay, you are not going to get two unequal strands like equal, right? In this case, equal. Both are equal. So this is called as blunt end. The blunt end, main drawback of production of this blunt end is it will not produce sticky ends. If sticky ends are not produced, obviously it will be very difficult for the ligase enzyme to join them. So we need extra procedure for it. What is this extra procedure if blunt ends are produced from this particular restriction enzyme? There are so many restriction enzymes which will produce blunt ends. Okay, so we need for extra procedure, that is nothing but addition of adenosine and addition of thymine, okay, to the vector DNA as well as the foreign DNA. So I have to, this is nothing but poly A and poly T. Got it? Where exactly? To the terminal end of this particular DNA. So I am going to add, so I will use pink color, so it will be easy. A. It could be like, I am not sure about like how many A's will be added. Here this strand T, that is automatic. Okay, and in this case we just add T. In this case A. Okay, 4 A's. This is nothing but addition of A and T to the blunt ends. So this is the procedure to be done in the laboratory manually. Okay, it is not automatic. Okay, it is manual. And after that, okay, now once this poly A and poly T process is done, what we have to do is we have to join these two DNAs to make recombinant DNA. For that we need one enzyme called as terminal transferase. This enzyme is required only in the case of blunt ends. Okay, so this terminal transferase is actually very helpful in this addition of this poly A and poly T tails to the blunt end DNA. Once this is done, we need to treat these two different DNA with ligands. So what is the function of terminal transferase? They help in poly A and poly T process of the blunt ends. That's it. So this is the thing to remember. Don't get confused with the sticky end and blunt end. Both are entirely different. Okay. I want to add one more point here. The cohesive ends can also be called as staggered ends. So please note all these points. Okay, no confusion here. Cohesive ends can also be called as staggered ends or sticky ends, not as blunt ends. Blunt end is entirely different. Hope you have understood this. Okay, no confusion here. I was a bit confused here. Very simple. For the process of these two, poly A and poly T, we need this enzyme because they are going to add A and T to the blunt ends, this particular terminal transferase. After that, once the process is done, we need to treat these two DNAs with ligase. The procedure is same. Okay, so that's it. That is the difference between sticky ends and blunt ends. Okay, so hope you understood this. Please write down each and everything. Very, very important. No confusion, nothing else. If you have any confusion, please call me. Okay, thank you. Now the last schematic representation, again it's very important, diagrammatic representation of recombinant DNA technology, again very important. Okay, simple, very easy. What we do is we just draw the vector DNA here, and here we draw our gene of interest or foreign DNA. You have to write full FDNA, and this is VDNA or vector DNA, VDNA. Okay. So here what we have to write is treatment with restriction enzyme. Same here, treatment with restriction enzyme. Copy the same. Okay. Once this particular vector or the foreign DNA is treated with the restriction enzyme, what you are going to get is you are going to get this. Simple. Simple, very easy. Use common sense. Done. In this case, you are going to get the fragments. See. Okay, fragments will be available. So just write down fragments of foreign DNA. Here nothing. You just write the vector DNA. Okay. So once it is done, the process of ligation takes place. That is nothing but with the help of the ligase enzyme. Don't write sticky ends or blunt ends. Nothing. Just write ligase enzyme. To join treatment with the ligase enzyme to join vector plus foreign DNA. That's it. What you are going to get is you just write down this. No need. I mean, if you want, you can just draw a double-stranded DNA. But in this case, single strand is enough to understand. Recombinant DNA is done now. We have got the rDNA. And the next step is, I cannot continue further, no space. So what I am going to do is introduction again. Introduce this rDNA into host. Right. Simple. Okay. So we just draw host. It could be bacteria, human cells, whatever it is. So we just try to draw this. Done. Okay. rDNA. Then next is cloning. What is cloning? You get multiple copies of the bacteria or the host cell containing multiple rDNA. Done. I think you can draw it. Yes, right? Easy. Very easy, right? Simple. That's it. Okay, hope you understood this. So this is actually diagrammatic representation of rDNA technology. Thank you. Please watch all the videos. Do not leave the videos. Got it? You have time. Okay. Every day at least spend 1 hour for this biology videos. I'm going to complete the syllabus as soon as possible. Okay. So don't be lazy. Done. Study very well. Thank you.